Reed. I'm Dean of the College of Health and Human Performance, and it's my very great pleasure to welcome you to this event. This is the installation of Dr. David Vaillancourt as the endowed Orchid Chair in Applied Physiology and Kinesiology. A very unusual and exceptionally happy event. We're delighted that all of you can be here. I want to begin by thanking the organizers of this event. A lot of work has gone into this. So Michael Spear, who's our Director of Communications, he's with us today. Also from his office, Bridget Welch, Jeremy Clark were instrumental in this. Michael Murphy is the guy who's our facilities person and took care of so much of the logistics here. And Melissa Cunningham is working to keep the reception going and get it organized for after the event. And so you have her to thank for that. I also would like to acknowledge the people who we are so pleased to have here, including Dr. No, I won't call Keith beyond name. We're delighted to have so many students in the room. Students need to see this part of academia. This doesn't happen very often, the installation of an endowed chair. This is a very special time. The staff of the department and elsewhere throughout the college are here with us, and so many of Dr. Vaillancourt's colleagues, the faculty. I'm especially glad to see Professor Tucker, Professor Clanton, two others of our endowed faculty holders. In addition to that, there are many distinguished guests in this crowd, most notably Dr. Vaillancourt's lovely family and these folks who were down here. You saw them in front. Such a blessing to have them back with us again. It's also great to have David's friends from off campus and many of Dr. Vaillancourt's colleagues from the medical center and elsewhere. Thanks to all of you for joining us for this event. I also want to take special pride in acknowledging Catherine and Brian Carwile. Catherine and Brian are here. Would you stand up, please? Catherine and Brian are the people who had the vision, who had the commitment, who had the capacity to endow this chair for this university. They have, by doing that, made an ongoing impact that will affect generations to come. Dr. Vaillancourt for many years, his successors, those who follow in his footsteps, for generations, there will be an endowed chair holder for the Orchid Chair, and it's due to their generosity that we're able to do this. Let me take a second to talk about what an endowed professorship means, and this is more for maybe the students than anybody else. The idea here is that you got to understand, and I think on an academic campus it's a little hard to realize that actually PhDs are a rare bird. Maybe 2% of Americans earn their PhDs. Among those few people, maybe 3 to 5% of the PhDs ever attain tenured professorship. So that goal by itself is very rare, it's very unusual. However, within that population, there is one more step, and it's not something that a scholar can attain by their works alone. It's a partnership. To be an endowed professor or an endowed chair means that an endowed position must exist. Those are few and far between. So to be an endowed chair is to reach really the highest academic level of honor and respect and distinction that the academy can offer. So that is what we are celebrating now, and we are honored to have David Vaillancourt in that new role. At this point, I'd like to turn the microphone over to somebody who's much better spoken than I am. This is Dr. Steve Coombs. He's a tenured professor from our APK department, and he will do the introduction of Dr. Vaillancourt. Steve, please.
Thank you, Dean Reed. Can you hear me at the back? Everybody's good. So it's a great honor to do this. For those, I look out in the crowd and I see a lot of people I recognize, there's a lot of people that I don't. And so my, I guess, history with David goes back to when I was doing my first postdoc. This was back in 2005, 2006. I went to a conference, I saw a guy speak, and I'm like, I need to work with this guy. I need to figure out what he's doing, and I need to learn how to do that. And so then it was like November 2006, I started writing the email of how do I approach this guy? How do I figure out how I get to go work with him? So I wrote this email, I had my wife check it, I wrote it, I checked again. It's like, this is, you know, you go back to like, this is kind of how it was, right? You knew what you wanted to get. But anyway, I sent this email off nervous, and it was probably within a day or two I get the email back. As we all know, David now he's on it. You know? I get the email back, we're talking on the phone probably within a few days, and then it kind of began. So then during that 2 years, ended up collecting data up in Chicago to write a grant. And then another thing that I've come to know very well is it turned out that David hit his grant before I hit my grant, which ended up funding me on a postdoc to go work with David. And then we've basically been working together since 2008. So it's kind of one of these funny things that you think, like other than my family and my kids, the person I've probably spent the most time with is David, which is, you know, which is a sobering thought anyway. No, it's a great, yeah, it's been great. It's probably one of the best decisions I made, I think, to work with David. So what I'm going to do now, I have maybe about 10 minutes to give you kind of an introduction on David's career and why I think he's so deserving of this great work. There we go. All right, so the title here, The Progression of David Vaillancourt. And that will make more sense when he speaks about the work that he's been doing, looking at progression markers in Parkinson's. But this is going to be the progression of David's career. So he got his undergraduate at Texas A&M back in '97, different century. One of his professors, Dr. David Wright, who was professor of kinesiology, suggested maybe he should think about research, which was a phenomenal tip, I think. And so based on that, sorry, I'm going the wrong way. Based on that, he then went to Penn State to pursue a master's degree and then a Ph.D. With Dr. Carl Newell. And during that time developed, I'm going to use the word simple, but it's not easy. It's simple experiments that allowed him to basically track activation in the motor system. And at this point it was using what's called EMG, so kind of studying muscle activity, and then designing really precise experimental manipulations to basically look at if we change X, what happens to the way we move. And so based on that early work, so he was there for 4 years. He then transitioned to a postdoc in Chicago with Dr. Daniel Corcus, and basically worked there on studies with deep brain stimulation with Parkinson's patients, again looking at how that changes motor symptoms and the output basically of how people move. In addition to doing that, he wrote his own first NIH grant, an F32 grant, and was basically interested in functional brain imaging. And so this is one, as I've kind of gone through this and you look back, you see these inflection points where trajectories kind of changed. And this is really the first one where he basically wanted to know about brain imaging and how the brain is controlling movement. And so developed these really nice paradigms that you could run in MRI scanners where you're basically assessing how the brain is controlling movement and having people move, and doing it in an MRI scanner where the one thing you're told not to do is to move, right? So it's kind of there's a challenge to this. And actually this paradigm, this was published in 2003, and this paradigm has now been used across the world, many different groups, partly because it's so simple, but was hugely challenging to get it going. And so the early work was in healthy controls, and then that transitioned to patients with Parkinson's disease. So once you have these models of how to assess brain function, you can then start rolling that out to understand what happens in different disease states, which is exactly what David did. The other funny thing, when I go back through this, some of these papers, so this was in 2007, and I actually went back and I read the paper, and there were 7 subjects in this study, which for anyone in science, in human, like 7 subjects, you're like, really, they published that? But then what you realize is the reason they publish it is because this was at the cutting edge of what was being done. Nobody was doing this with Parkinson's patients. with functional imaging in the scanner. And it turned out that even with the 7, it actually is held, right? So that's the other thing with science. You hopefully do good science and it gets replicated by others. And if it does, it must be true or close to true. If not, then it falls away. And so what you see in David's career is time and time again, he's published work that's basically stood the test of time. Another point, so there was the one inflection point of the functional imaging. This is, I think, the second one. So this paper was 2009. One of the things I've seen, working so closely with David is his ability to adapt. So, you hit a grant, you publish a paper, that's great. Maybe it's good and you just keep rolling on the same thing. You just kind of bleed that dry, right? David's philosophy is very different. You hit some success, you leverage that to go in another direction, take another big swing. And so what this data is showing, this is what's called structural imaging. And this again was in 2009, there were 20 papers published using structural imaging in Parkinson's disease. About 5 of them were data-driven, and 1 of them was David's, and it was the best paper that was basically published. Again, right at the front end of this field developing on looking at biomarkers in Parkinson's disease. And using structural imaging is huge because clinically it's more available than the functional. And so again, rather than kind of just enjoying the success of the functional, you kind of move towards the structural as well, and I think what he's going to talk about today is going to be where this has gone over the last 15 years. So you can see here, this is kind of the progression. What you'll see, assistant professor in 2005, tenure and promotion in 2009, so 4 years. Typically this is going to take you 6 or 7 years, so we're already fast tracking. He was then recruited here to Florida in 2011 as associate, and then to full professor in 2013, so again another 4-year gap. Basically fast tracked, right? And you don't fast track because people like you, well, partly because they like you, but partly because you're a good colleague, let me get my words right. But you've got to be producing at a serious level, right? You've got to be at the very top of the game. So this idea of an endowed professorship, I think there's nobody more deserving than David. So then he became a chair in APK in 2019. So I wanted to talk a little bit about the science, but then now I can give you the metrics. So this was the first part of David's career from 2000 to around 2011, publishing prolifically for a junior investigator. And then, so what you can see here in purple, these are the number of papers, and then in blue, that's the number of citations. What that means is that's the number of other scientists that are referencing David's work, right? And you can see that is on a steep incline. And so then David and I started working together. Did I say that out loud? So what happened basically in 2011, David came to Florida, came to an environment, see Dr. Open here, Dr. Bowers. What happens when you take a good person and you put them in an even better environment? Basically, this is what happens, right? You see the publications go up, the citations go up. And so the numbers on the right, you can see over 200 publications, which is huge. And again, the impact of those publications is massive. It's been cited by other researchers 8,500 times with an H index of 53, which basically means he doesn't miss. The quality and integrity of this work is just solid. So the other thing, for those of you that know David, we don't really talk about money, even though it kind of is sort of about money, but it's not really about money, but it's helpful, right, to do the science. And so grants is a big thing, and I wondered, do I put this in? But it's legendary status, so David's ability to get grants is phenomenal. And so what I've done is overlaid some of the early grants that he got, which these are kind of good milestones in a career as a scientist. And then it gets, again, ridiculous. And I work with him every day, and I didn't even realize I'm looking through his resume. I'm like, you've got to be kidding me. It's like another one I didn't know about this one. It's amazing, and this is important partly because the money allows you to do the science, but it's important because if you get money and you get it early, maybe you're lucky, maybe it's not lucky, but if it's lucky, most people don't go on to get another big grant, right? That they hit that one and then it kind of slows down. What this shows is if you give David money, he's going to produce, right? And then he's going to write another grant that's going to keep producing, and he's going to keep going and keep going. And I feel like that's one of the things, that the relentlessness of the pursuit of science is just extraordinary. It's awesome to be around. But not everyone's a winner, right? And so what's amazing here, so as academics, we write grants as the principal investigator, as the PI. I had to go to 2013 to find the first grant that David wrote that didn't get funded. I can't even begin to explain how phenomenally hard that is. When these grants are funded, maybe 10%, the 10th percentile, and he didn't miss until 2013, which I remember. But yeah, it's an extraordinary achievement. The other thing I want to say, so I was kind of asked to talk about his contribution to science, how the impact that he's had on the field, and so from that there's the scientific part, but there's also the training part, right? One of the novelties of being in academia is we train students, we train postdocs, and then in a sense your legacy or your reach goes beyond just what you're producing. And so on the right here, these are the people that David trained with, and then we have junior faculty that David mentored, and then here in the middle, these are all postdocs that David's worked with, and then on the right, Ph.D. students. So it's a big family of great people, and then the impact, the colors are a little off here, but basically everybody that just changed there are all still doing science and still impacting the field, right? And so that's, again, it's kind of this measure of a good mentor, a great scientist will train great people who go on to kind of, it's almost like compound interest, right? If you train good people, the legacy grows. And so as my final slide, so I actually reached out to one of David's previous postdocs and told her that I was doing this, which she was very excited about, and then told me about this. So there's a website called Expertscape, and basically you put in David's name and it pops out that he's in the top 0.13% of experts in the area of Parkinson's disease, which again is pretty cool. It's kind of, it's consistent with everything we know about what he's doing, but to see the metrics is great. And so my final point, like, is again, I've been going through this, like, what's kind of the take, what's the magic behind this, right? If you could bottle this, whatever it is that David has, what would that be, and what would it look like, and how could you kind of leverage that to basically keep going? And this is what I came up with. So now let me present Dr. David.
That made me laugh. Can everybody hear me okay? Is the mic working okay? Yeah, I started to figure out, I think that's where Steve's going, and the Dave Wright picture sort of set it off to me. So I want to thank everybody for coming. I see a lot of really good friends and colleagues, and it's just warms my heart to see so many people I care about. So I just want to say thank you very much. I want to say thank you to Steve for such a kind introduction. Thank Dean Reed for his support. And I would like to tell you a little bit about what motivates me and motivates me to do science. So what motivates me is my family. My wife is, she's my best friend, and I'm on this journey with her, and I'm very, very thankful and happy that I'm on this journey. We also have three wonderful daughters. Sydney, Abigail, and Lily are here. This is them when they're younger. And they typically do grow up. They get more beautiful and capable as they get older, and it's just been wonderful seeing them grow. And that is my why. That's why I do what I do. I also want to thank and acknowledge our laboratory. Steve is up in the upper right, but our lab has had so many different people come through the lab. This shows you a picture of current people in the lab, but also previous people in the lab. And we've just been so lucky to have people come through the lab, and I'm just taken aback by them, and I appreciate all of them. This is a picture taken to thank Becky and Bob Allen, who couldn't be here today. They donated money to our lab a few years ago when there was a flood. There was a sewage flood that came in during the Florida-Alabama football game that put about a half a foot of water into our lab. So thankful to them for putting our lab back together. But most of all, I want to thank Brian and Catherine Carwile. They are the people who we are here today because of them. They put together this vision, and they put together the impact to help the college in the university going forward. And so I just want to say thank you to them from the bottom of my heart. And this is Brian in front of our dean's car. So. So this is one of the oldest videos that I know of on record for Parkinson's disease. This shows a patient, and you see all the symptoms that are sometimes masked by medications or deep brain stimulation these days. You don't see patients like this. As you see a stooped posture, you will see a rigidity of the patient, you'll see slowness of movement, you'll see tremors, and this is really what patients experience when they're not on medications. So you typically are not, you will not see this too often today because the medications they take, the dopaminergic medications, can many times sort of mask these features. One of the things that I think is a hidden aspect of Parkinson's disease, and I have many neurology colleagues in the audience, and they will tell you this first up, is that the diagnosis is not as accurate as you might think it is. So if you look at pathology, which is the gold standard, and you compare that with clinical diagnosis, in the first 5 years of a clinical diagnosis, it's roughly 55 to 75% accurate. That's what the field tells us. And so standard of care right now is if you go see a physician, well, they may or may not order an MRI. That's probably, they may or may not order it. So the only reason they would order it is to rule out vascular Parkinsonism, maybe a stroke or a tumor, but they're not using it proactively in diagnosis. So we've been using a technique called diffusion imaging, and I want to tell you a little bit about the basic physics of diffusion imaging. It's looking at the way hydrogen within water is diffusing in the tissue or in the body. And so basically, if you have unconstrained diffusion, the hydrogen or water molecules are basically going to diffuse unconstrained. But you can have constraints in the brain. So you have cells, you have tissue, you have gray matter, white matter. And so if you have some sort of constraint, that diffusion is going to go along a specific vector. And in diffusion imaging, what we're doing is measuring the microstructural environment within a voxel that teaches us a little bit about the diffusion process going on. And changes in the biology of the patient or in the disease process will alter that diffusion mechanism. So this is a voxel. We use what is called free water quantification. This is a technique that was originally developed by Pasternak, who's an applied mathematician, in a paper which was published in 2009. And we've been using this technique largely because I think it has enormous clinical applicability. It's scalable to clinical worlds, and it can be used on many different scanners. And what I'm going to try to show you today is where we're at in that process. So in free water imaging, it's basically assessing a voxel, and the diffusion is going on in two different spaces. One is the extracellular compartment, which is in blue. That's outside the cells, and that's the free water compartment. And then the red is diffusion within the cells. And whenever we're quantifying free water imaging, we're quantifying two metrics. One is free water, the fractional volume of that extracellular compartment, and two is the tissue constraints on the diffusion process as well, basically what's going on within the tissue itself. So early on in my career, as Steve said, we focused on the substantia nigra. The reason we did that is because that is the hallmark pathology in Parkinson's disease. It is the part of the brain which loses dopamine and stops producing as much dopamine, and it's the part of the brain that is the most consistent area at postmortem for a patient who's passed away. So a pathologist would be looking to see if the substantia nigra has changes. And in doing so, what we wanted to do was image the substantia nigra and see if we could sort of see things that might teach us a little bit about how a patient is doing and teach us a little bit about how a patient might be progressing. So we used this technique in 2015. This is the first sort of study to basically look at the progression of Parkinson's disease. And this is the first progression marker using MRI that was published in the literature. This is done by Ed Ofuri, who's now on the faculty at Arizona State University. But what Ed did was basically identify in the midbrain region, you see yellow and white, that means low free water. High free water is seen in these blue dots. Those blue dots is the substantia nigra. And so that high blue means high free water within that structure. So this is no contrast, there's no drug injected, and we're basically able to sort of image that structure and visualize and quantify the substantia nigra. And so what we did was we found that basically this is the patient at baseline, this is the same patient a year later. You see the substantia nigra intensifies, it's the free water content, and it's growing, it's getting worse. We then followed here at the University of Florida a group of patients with Parkinson's disease and healthy individuals, and what we found is that the controls don't really change over 12 months, but the PD patients increase even further. And what that said to us was basically that the substantia nigra is degenerating progressively over time. And that was the first demonstration that we saw of that in the literature. And that finding has now been replicated by many other labs, not only across the US, but across the world, and it's also been replicated by our laboratory as well. In parallel, we wanted to start studying what goes on mechanistically within the substantia nigra, or what goes on mechanistically within the brain, which might sort of alter these free water signals. So we started studying animal models with a colleague named Ben Giason here at the University of Florida. And what UF has, which you may not know, is something called the National High Field Magnet Lab. There's wonderful high field magnets which provide a wonderful facility to do high field imaging and do really cool science and neuroscience experiments. So Winston Chu did this in his dissertation. We basically used a model, which is Ben Giason's model, where they inject preformed fibrils, alpha-synuclein fibrils, into the hind limb of the mouse. And that basically works its way to the brain within about 3 months. And then within about 3 months, the mouse will have rampant synuclein pathology throughout the brain. Now, synuclein itself is thought to be one of the hallmark pathologies of Parkinson's disease. So in these experiments, we're doing longitudinal imaging and trying to understand when is diffusion imaging changing relative to the synuclein pathology. And what we found was, we found a couple of really important things. First, we found that synuclein is actually inducing diffusion changes in the brain, and we found that the changes in diffusion imaging that we see are in similar regions of the brain as alpha-synuclein pathology. So that suggests a link between the signals that we measure quantitatively and the pathology of the disease process itself. Second, in a series of other experiments, we wanted to look at other mechanisms of cellular decline where we looked at inflammation and also neurodegeneration. What we identified was that both of these mechanisms will alter free water signals. And it sort of makes sense that as you lose cells or as tissue is dying, water is going to take over that microstructural environment. But the other side of that coin is as inflammation is occurring without atrophy, you'll also see increased in free water signals. So the other kind of bit about this as we went forward is that I started listening to people who would tell us how to use this technique. And so several different people in the field, when we had our first publication, started telling us we should start looking at the types of Parkinsonisms. And so Parkinson's disease is not necessarily just one disease. There are other forms of Parkinson's that can get misdiagnosed. And that's the reason that in standard clinical care, the misdiagnosis is so high. And that's because when a patient presents to a physician, the patients look and present symptoms that look similar to other patients from another pathology. And so that misdiagnosis rate is just basically due to the fact that it's difficult to differentiate these patients early on. And so these are some of the most famous faces of Parkinson's that we would all probably recognize. And they probably all have a Parkinson's disease diagnosis, but it's also possible that they could have other pathologies that they don't know about. So one of the things we started to do was realize the power of machine learning. And so if you have a phone, I actually have an older phone that still has a thumbprint on it. And so one of the things it does is it reads the folds of your finger, and it uses some sort of machine learning model to fit to my finger. And if it reads my finger, it will hopefully unlock the phone. If it reads one of yours, hopefully it will stay locked. And so by the same token, we're using machine learning to read different parts of the brain and then develop a model that we can trust and then see if it predicts the disease. And it's basically the same idea as unlocking the phone. So when Ed published that initial paper, one of the things that happened was that people started emailing Ed. They didn't email me, which is kind of funny, and they wanted the free water algorithm. And I said to Ed, it was like, no, we can't share that, Ed. That's not possible, because Ofer told me initially you can't share that or send it out. So we started telling sites, well, send us your data and we'd be happy to analyze the data for you. And what that did was over time it helped us accrue a data set. And we started accruing a very large data set, and I think it's probably one of the largest data sets in the world. And so in machine learning, data is kind of like water. You know, having control of water is useful. Well, having a lot of data is useful in machine learning. That's the most useful part. So we put together a paper in 2019, which is really a proof of principle to see if this can actually work. This was published in Lancet. This was done by a student of Steve's, and he was a postdoc with me named Derek Archer, really a fabulous person and scientist. And so what we had was over 1,000 participants, 8 international cohorts, 17 different MRI scanners, different scanner vendors. This is a real-world messy data set. Nothing was matched, nothing was harmonized in data collection. So really was challenging because we didn't know if it would work. And what we were able to do was in the study, we were able to differentially diagnose patients with over 90% accuracy between different forms of Parkinsonism. Namely, we looked at Parkinson's disease, and we also looked at 2 other pathologies, multiple system atrophy and progressive supranuclear palsy. These other pathologies are ones that are more frequently seen clinically and more difficult to diagnose in comparison to PD. Now, this is a retrospective cohort, and so it doesn't mean that it's going to work in practice. And so some scientists would stop here, but I'm telling you, this is not where you stop. You need to actually test it to see if it's actually going to work in practice. So one of the things I did was I reached out to 2 good colleagues, Michael Oken, who's here, and Inglis Barmpoutis. And I first went to Michael and I asked him a little bit about, how would we do this? What would we do? And his suggestion was like, you got to go to the Parkinson study group. So I approached the Parkinson study group, and we started asking questions about how we might sort of enact this particular study. And what we did was we asked sites how they would want to get involved. We sort of vetted the sites and came down and narrowed the sites down. Now, Angelos, in this case, is a computer scientist, and he was able to sort of build the infrastructure to allow sites to start sending us their data in an IRB-friendly way. And that was truly important at the time. And we didn't realize at that time that what he was developing would actually become potentially clinically useful. So we have been running this study. I can't show you the results today. We have tested 320 people. We've tested them across the sites that you see right here. 19 sites in the United States and 2 in Canada. It's a prospective study, so we're using previous data that we had, and we're training the models and taking a little bit of the new data and seeing if we can test it prospectively. And then we also have a cohort of pathological cases where we're also trying to ask the question, can we predict pathological cases from a completely different cohort of patients? This just shows you some of the people involved. So Marissa Schauder is our study coordinator. She has, I would say, single-handedly sort of gotten all these sites to do the work that involved and gotten 320 patients tested across these sites. Sam Wu is our statistician involved. Rogu Feng is a machine learning expert. Stefan Prokop is our neuropathologist. Wei Wang is in the lab and is an image processing expert. This is Rob Chen, is a Ph.D. student, really responsible for doing 99% of the image analysis. And then Todd Parrish is an engineer who's helped set up the sequences at the sites. So pretty soon, we're actually working on the manuscript now, and we'll be sort of sending it out for publication in the near future. So I don't know if you remember, but a few years ago, a pandemic hit our world, and we were all sort of put into our houses for a period of time. And one of the things my wife did was put on our computers while we were at home was these little signs. And so this is on my computer now. It's still there. And it tells me to focus and say no to distractions. And she did that because we were at home and it was very difficult to say no to distractions. And I'm sure you guys experienced similar things. But I used this to sort of drive me going forward. But during that process of going through COVID, it started to also make me think a little bit about how I might impact the world. And so when you publish papers, it's important and very important, but it doesn't necessarily take that next step. And that next step is really translating academic discoveries into something that can be used for patients. And so I started realizing that, well, to do that, you actually have to start a company. You actually have to develop a commercial product. So I was also started talking to a number of people at the university. Angelos and I did. He's my business partner. And we started getting a lot of advice from people at the university. So there's a wonderful technology licensing parts of the university. And they started teaching us a little bit how to do this and what to do. And so in 2021, we started a company called Neuropax, which is basically trying to commercialize this particular diagnostic software for Parkinsonisms. And so we've done a few things which have been good. We took some training initially. So this was actually during some of the COVID stuff. We took an I-Core training. We took a training course to teach us how to write small business grants. So we were able to get a couple of grants. We got a grant from the FDA, a small business grant from the NIH. And we've sort of taken this to the point now where we're submitting for what is called de novo pathway submission to the FDA. So when it's done, we think it'll be done by the end of this year. It'll be hopefully approved as a Class 2 medical device, which can be used clinically. We've been following the advice of many people teaching us how to do this because we seriously don't know what we're doing, and we're trying to figure it out as we go. In addition, we're applying for what is called biomarker qualification through the FDA, which would allow it to be used in sort of pharmaceutical or device trials as a diagnostic device. So this is the, not the actual product, but it kind of gives you an interface into what it is. And so basically what happens is, this is the front end, which Anglos has created. And so it's basically interfacing with the server, and so data can be dropped in, and then processing starts to occur. So it takes about 2 hours right now to process a data set. We think we can find ways to make that faster, but we haven't started doing that yet. And in doing so, it's basically doing the image processing, it's making sure everything's sort of accurate, and then eventually it's going to start doing the machine learning as well. And then at the end, it's going to provide like a diagnostic type report. So this shows you an example here. So this is a patient that would have MSA, because you can see the MSA is likely there. In a different case, you might have a patient who has Parkinson's disease, which is shown here. And so the diagnostic report right now is a classification, and then down here it shows some of the key regions of the brain that might be driving that classification. And for anybody who's worried, we have serious conflicts of interest that are overseeing all of this, and I can assure you Anglos and I are following every rule at the university. So last, I wanted to touch on a couple of future projects that I'm sort of close to my heart and doing right now. One is studying things that happen before Parkinson's disease. So most of my career has been focused on after diagnosis and trying to understand what's going on once diagnosis occurs. But one of the things I'm trying to do now is start looking before diagnosis. So sort of look at cohorts and at-risk cohorts where we might start seeing if we can develop biomarkers that might occur well before a patient is diagnosed. And the reason for this is because I'm pretty much convinced that disease-modifying therapies that might slow the disease process need to be done before clinical diagnosis. I'm not the only one convinced of that, but I think the field at large is starting to really kind of recognize that. And so most of my work or thinking about grants in the future is really focused on doing science before a clinical diagnosis. So we're doing a study right now in what is called rapid eye movement behavior disorder. This is a disorder, it's a sleep disorder where people will act out their dreams at night. So when you're in REM sleep, basically your muscles are very quiet and you're not supposed to move. But during REM sleep, patients with RBD, or rapid eye movement behavior disorder, actually start contracting muscles in an involuntary way. And those patients go on to have a very high risk of developing Parkinson's disease. So about 50% of those cases go on to get Parkinson's disease. So we're studying that cohort right now, trying to understand if some of the markers that we've been studying, potentially other markers as well, might show up very early on. In addition, this work has been done with Mike Jaffe, who's in neurology, and also Evangelos Christou, who's a colleague and good friend. Finally, for personal reasons, but also scientific reasons, I have a very deep interest in Alzheimer's disease and dementia. Our lab has been playing an important role in what is called the One Florida Alzheimer's Disease Research Center. This is a network of universities put together to study Alzheimer's disease within the state of Florida. So it's University of Florida, University of Miami, Mount Sinai Medical Center, FAU, and also FAU are involved in this. Our PI is Glenn Smith, and good colleagues would be Melissa Armstrong, and this is a postdoc, Jesse DeSimone, who's working quite a bit on this project as well. So these are areas that I'll be looking at going forward. And finally, I just wanted to say a shout out to Dawn Bowers, who's a good colleague. She and I have been running a T32, which is a training grant for Ph.D. students across the University of Florida. These are some of the pictures of patients, of students who've been involved in the program. Some of them come from our college, but most of them actually come from other departments and colleges across the campus, from engineering and neuroscience and physiology and clinical and health psychology and speech and language pathology. Dawn and I are, I think we're a very good team, and we love working together. And soon, Melissa Armstrong is going to be taking over for Dawn's roles. So I'm very much looking forward to working with Melissa on this project as well in the future. So as I kind of close, I just wanted to turn it back to the reason we are here today. I want to thank Brian and Catherine for everything that they've done, and I look forward to to hanging out with you again in the future and spending as much time as possible, and I really appreciate everything that you've done. So thank you all very much for your attention.
Yeah, so for those few of you who did not know what this man does, that gives you a little glimpse into why we are so proud to have him be the inaugural Orchid Endowed Chair in APK. David, I don't need this. We have created a medallion for you to honor you and to honor this new endowed professorship. On one side, it has Orchid Medical Chair, and on the other side, I believe it has a little information about you. So again,
Thank you.
congratulations very much. We're so proud of you.